everyone. I am Dibyendu Samanta from IIT Kharagpur, and today I will be discussing about transcription, the involved machineries and processes. This is the second lecture of module 2. Here I will be covering this topic like overview of transcription process and then initiation of transcription followed by RNA polymerase and the mechanism of RNA synthesis, and finally how transcription stops. So in this, I would like to first give you an overview of transcription process. So as I already mentioned in the previous lecture that we have around 20,000 genes, protein coding genes in our genome. So now in that respect, I can say that in one DNA molecule or in one chromosome, so we have multiple genes. So for example, if I say that this is part of one DNA, like double-stranded DNA, and since DNA are anti-parallel those two strands, so I am just putting here something like this, and DNA can be very, very long, and one DNA can have multiple genes, like 500,000, something like that, the number of genes present. So now I would say this is one gene that can code a protein, and this is another gene, this is another gene. So something like that, it is present throughout our DNA molecule. But now, as we can see, for example, in our body, so we have nerve cells. The function of nerve cell is completely different from the function of liver cell, and their shape, size, morphology, that is also different. But now if we see their information, the DNA content present in liver cell or the nerve cell, the information is same. So that now the question is how then they are doing different function, why they are completely different in terms of their shape and size, everything. So this is because of how those informations are being processed to make the product. So now here, in order to process the information present in this gene, for example, I am just naming that this is gene A, how we will make the product from this gene? So the idea is here, if I just show this portion, like just I am forgetting the rest of the DNA here now, so I am just mentioning here. So this is my portion of this gene. So here we have the gene A. So in order to start transcription, that means in order to make or initiate the RNA, we have to understand from where we have to start, because like most of the DNA or most of the nucleotide, they are not transcribed into RNA. So as a result of that, something should be there just ahead of the gene which will initiate the process. So here I would like to mention that we have some specific sequence before the gene. So this sequence is called promoter sequence, some kind of specific sequence present here. So this promoter sequence will invite RNA polymerase, right? So as a result of that, this promoter sequence will invite RNA polymerase. This is the enzyme RNA polymerase. From the name you can understand, so just like DNA polymerase, so this is also making polymer, but polymer of ribonucleotides, and that's why this is called RNA polymerase. So now compared to replication, this is much more, I would say, simple, not that much of topological and logistic problem present here. So we don't need helicase, topoisomerase, lot of enzymes, because this RNA polymerase alone can break double-stranded DNA just in a small part, so that we can have access to single strand and we can make the RNA. So here, if I go to the next step, it looks like something like this. So I would say this is 5 prime, this is 3 prime, and 3 prime, 5 prime. So RNA polymerase is present somewhere here. So this is our RNA polymerase. Now, the synthesis of RNA is just similar to DNA synthesis, like it will always grow from 5 prime to 3 prime direction. So as a result of that, I would say that whatever sequence present here in the template, the RNA will be synthesized in this way. For example, in this DNA, if I say the sequence here is A, T, G, C, C, A in this strand, and the complementary strand sequence should be here T, A, C, G, G, T, and we can continue like this. So that means when the RNA is being synthesized, this is template directed synthesis. Whatever present in the template, it will make just complementary sequence. So as a result of that, in this RNA, what will be the sequence? So here, instead of A, T will be coming here, but T is not present in RNA. RNA polymerase will not recruit T. Instead of that, it will have U in this RNA, and then A, C, G, G, U. In this way, this RNA will grow. So now if we see the sequence of this RNA which is being synthesized, this sequence of this RNA is similar to this sequence, exactly similar to this sequence, right? And complementary to this sequence, because in this case, this strand of DNA is acting as template strand. So that's why we are getting the complementary sequence here, and which is similar to this strand, and that's why we say this is template strand and this one coding strand or sense strand, because whatever code or sense present in this strand of DNA, it is coming into this RNA, and that's why this is called sense strand or coding strand, and this is template strand. So I will discuss in more detail in the next slide. So here, this is just the RNA polymerase is synthesizing this RNA from 5 prime to 3 prime direction, and then we should have some mechanism so that this will, like RNA polymerase will understand that the gene is over. Otherwise, it will transcribe the whole DNA, but that is not our goal. So as a result of that, some stop signal should be there also, just like a promoter sequence where this process started. Similarly, there should be some stop signal, and therefore the RNA polymerase will stop there, and RNA synthesis will be done, and RNA will be coming out from the from this complex. And this is like a little bit summary of transcription, and I want to discuss in more details about this promoter first, which recruits RNA polymerase, and followed by RNA polymerase, its action, how it actually help in RNA synthesis, followed by the mechanism of RNA synthesis, and finally how chain terminates. That means how transcription stop at right position. So as you can see here, promoter is just like a pointer that points to the location of the information, in this case particularly location of gene to be copied into mRNA. So this is, if I give you an analogy, this is similar to, for example, in a DVD player, like in a DVD. So we have, I would say, 100 different songs which I really like. And for example, today morning I I want to play a particular song, from this list. So, like whatever in that list, maybe the number 50 song I am really liking to hear today. So, I don't have to listen like number 1, 2, 3, 4, all these songs. I can directly select the number 50th song by some pointer devices, pointer mechanism. This is similar. Like based on the requirement inside the cell, we have very complex regulation mechanism. Cell will guide properly so that the particular gene will be transcribed. So that's why genes, like before the gene, we have the promoter sequence, and it will help to start the process. So as you can see here, that in this sequence, whatever we are writing here, like double-stranded DNA and 5' to 3' sequence we are mentioning here. So here the starting point of the gene, as you can see, this is the starting point of the gene, this A. So the starting point of the gene, the first nucleotide, we denote as +1 site. So I would say +1, then the next nucleotide T here, this is +2, +3, it will be gone like this. And just upstream, in this case on the left side, so we will go -1, -2, there should not be 0, right? So whenever the gene started, the first nucleotide is +1, and towards left side in this case, I would say on the upstream, it is -1, -2 position. So now what happened? In case of this gene, when it gets transcribed, as I just mentioned in the previous slide, for example, after transcription, we are getting this product, right? We are getting this product, this A, U, U, G, C is written here. Now, if you see this sequence is exactly similar to this sequence. Only instead of T, we have U here. We have U here. That's all. Otherwise, exactly same. So as a result of that, this strand, the top strand here, we would say this is the coding strand or sense strand. Whatever the information present, we are directly writing this information in RNA. So that's why this is coding strand. And in order to do that, we use this bottom strand as template strand. Sometimes we also mention like non-coding strand. So now, then where is our promoter present? In case of bacteria, particularly like prokaryotic promoter, approximately at -10 position, -10, that's why I mentioned which one is +1 and -1 site. So at -10 position, we have some kind of conserved sequence, some kind of consensus sequence. This is like called TA-TAAP. This sequence is kind of present in prokaryotic promoter, and it recruits the RNA polymerase, and therefore RNA polymerase will come here and then it will go in this direction, and finally we will have our RNA. So if it is like protein coding RNA, then we can say this is mRNA. So now the fundamental mechanism is same in prokaryote, in like bacteria, as well as in eukaryote, like in human, plant, everywhere. But the position of promoter, the sequence of promoter, that could be little bit different, that's all. But the basic understanding is same everywhere. So now, as you can see here, that the promoter sequence of eukaryotic gene is little bit away from the starting point of the gene. So this is around at -25 position. This is not just one promoter and some other regulatory elements present in DNA that is also contributing in gene expression or in transcription itself. We are not going into that much of detail. Now, here one important aspect I want to mention here, that transcription can start from both directions. What is that? So RNA, DNA, always they are getting synthesized from 5' to 3' direction. But since I discussed that coding strand, template strand here, so as a result of that, I want to clarify here, if this is one long DNA, right? So maybe here we have a few million bases, like few million bases present, like 5 prime to 3 prime and 3 prime to 5 prime, and few hundred genes are also present. So as a result of that, like whatever the way I just explained, that I would always say that this top strand is the sense strand and the bottom strand is the template strand. Template strand. No, not like that. Generally, what we see in textbook, always we see this kind of image figure here. You will be seeing that 5 prime, 3 prime, 3 prime, 5 prime, and the RNA is being synthesized in this direction. So 5 prime to 3 prime synthesis of RNA, and as a result of that, if this is the case, then here this is template strand and this is coding strand. But maybe this is true for this gene, this portion of the DNA, but maybe for the next gene this will be completely different. Maybe the next gene here present, and I would say transcription can happen in this way also. So this is 3 prime and this is 5 prime. So I would say here transcription can happen like this. So again 5 prime to 3 prime direction of synthesis. So as a result of that, what we would see? So whatever sequence here present, like A, T, G, C, and in the template strand of DNA, it should be here T, A, C, G, and in this RNA it will be U, A, C, G. So as a result of that, now this RNA sequence is completely similar to this, just the difference is U and T. So as a result of that, now this is the coding strand. Coding strand. So for this gene, coding strand is this one, that here. So as a result of that, that means the promoter sequence present here, whatever the promoter sequence we are telling that T, A, T, A, A, T, it is present here. Similarly, in this gene, promoter sequence, this T, A, T, A, T present here, and that's why it is happening. So what I want to mention here, in our DNA, it's a long sequence, but here information can present in both the strand. Like both the strand can be coding strand or template strand, depending on gene, its orientation and everything about that. So the system is very complex, but we have very good mechanism, we have very good machineries to read it properly and make correct product out of it. That's all. The next thing I want to discuss, that the RNA polymerase. I just mentioned that promoter sequence invite RNA polymerase in the site of gene, or just at the starting point of the gene. So as you can see, this is promoter sequence, and here the RNA polymerase. Now I would like to discuss little bit more about RNA polymerase. RNA polymerase is an enzyme, but now this enzyme can be little bit more complex. That means it has multiple subunit. So subunit means that multiple polypeptide, they can be together and they can function together. So that kind of structure is called quaternary structure of a protein, which will be discussed by Professor Soumya in next module. So that is called quaternary structure of a protein. So RNA polymerase is very complex, and as you can see, there are 5 polypeptide chain present in the polymerase, 2 alpha subunit, then beta, beta prime, and sigma. Those are just scientists given their name, that's all. So all these 5 polypeptides together, they make the RNA polymerase. And now I mentioned that RNA polymerase is getting recruited in the promoter site. Then how? So if we see the sigma subunit, this is sigma subunit of RNA polymerase. So it first recognizes the promoter sequence. So it has some affinity. Whatever the protein like polypeptide sigma, it can somehow interact with the promoter sequence, and therefore it first get recruited in the promoter site, followed by recruitment of other subunits like alpha, beta, and beta prime. And now this is the total RNA polymerase, right? This is RNA polymerase. RNA polymerase. Now, this total enzyme together is called holoenzyme. But now, in order to start transcription, the sigma factor should be released from the complex so that the RNA polymerase can move in this direction and it can make RNA out of this gene. Why? Most likely this is because that sigma, it has very high affinity towards the promoter site. So as a result of that, the function of sigma is to, that sigma factor or sigma subunit is to find out the location, like find out the promoter. Then other polymerase will identify, other subunit of this polymerase will identify sigma, and it will come and it will make this complex. But now if sigma get dissociate, then RNA polymerase can move. Because of this high affinity between sigma factor and the promoter sequence, it will be difficult to move this RNA move this RNA polymerase towards this direction. So as a result of that, whenever sigma will release, then RNA polymerase can efficiently move towards downstream of this gene, and it can make the product, it can make the RNA out of it. Now, a few important things I should discuss about RNA polymerase, that RNA polymerase is completely processive. So what is the meaning of processiveness? So that means our transcript is synthesized. Transcript means here the RNA, the result of the transcription, that is called transcript here. A transcript is synthesized from start to end by a single RNA polymerase molecule. So that means if this RNA polymerase start to transcribe this gene, for example, I am saying that this gene A, then we will get the RNA here. This is after transcription. This is, I would say, mRNA 5 prime to 3 prime direction, but this is being synthesized by single RNA polymerase. And here is the difference between replication and transcription. In replication, as you know by now, that multiple DNA polymerase take part to replicate one single DNA molecule. Particularly, as you can remember, in case of lagging strand synthesis, many DNA polymerase, they are being recruited to synthesize DNA in a discontinuous manner. In addition to that, another important thing about RNA polymerase is it can initiate the synthesis of RNA de novo. That means no primer required. Like during replication, we discuss a lot about the primer, that some enzyme specifically called primase that add some RNA primer, followed by DNA polymerase can synthesize the DNA from that point, from the RNA primer itself. But in this case, no primer required. That means RNA polymerase has the ability to directly start the process of transcription. That's why we are saying it has a de novo activity, de novo capability to synthesize or to start the RNA synthesis. And here, if you see some kind of statistics here, that RNA molecules present in E. coli. So if we see the total RNA molecule present in bacteria, particularly in E. coli, we will get that only 5% of RNA, they are mRNA or messenger RNA. So that means that code for directly code for the protein, and then rest of the RNA, they are tRNA and rRNA. Huge portion of the RNA is actually ribosomal RNA. So now, as you can see here, that who transcribe this huge pool of rRNA and tRNA? In bacteria, same RNA polymerase transcribes all these three types of RNA, but in case of eukaryotes, different RNA polymerases are involved in transcription of mRNA, rRNA and tRNA. So in eukaryote, we have three major type of RNA polymerase 1, 2 and 3, and they have specific function. Particularly RNA polymerase 2, it transcribes mRNA in case of eukaryote. Here some interesting fact, during the replication class I mentioned that Arthur Kornberg, he received Nobel Prize because of his contribution in understanding the process of replication. So he is Arthur Kornberg here, and here you can see Roger Kornberg. So Roger Kornberg is actually he is son of Arthur Kornberg, and again he contributed a lot in understanding transcription. So he also received Nobel Prize in 2006 for the discovery, or not, I would not say discovery, but contributing a lot in understanding transcription. So from the same family, one contributed in understanding DNA replication, and the next generation in transcription, and both of them they got Nobel Prize. This is really interesting. Now the RNA synthesis mechanism. So the overall mechanism is exactly same, that whatever I have discussed during the DNA synthesis, because of the 3' OH present in the sugar, in this case only this is ribose sugar, 2', 3' both are OH group, and from there the reaction start and the phosphodiester bond formation take place. As you can see in this slide, so this is the RNA which is being synthesized, and from 5' to 3' it is being synthesized, and we mentioned here template stand, and this is non-template stand, whatever I mentioned that coding stand, same thing. And now if we see the chemistry here, I would say this part, this is the growing RNA chain, this is this part. So as you can see here, growing chain, if I say the total number of nucleotide present is n number. Now in the next step we have here a residue, a nucleotide in the template stand. So as a result of that, one residue, that will be U, should be added here. So if I say this is U, nucleotide U, uracil, so it will be added here through phosphodiester bond here. So then the total number of nucleotide present in this RNA is n plus 1. So this way this RNA will be growing in this direction from 5' to 3' direction. That is all about RNA synthesis. Now the synthesis started, then at some point it should be stopped, otherwise it will be going and going. It is unnecessary making RNA out of DNA. So as a result of that, we have some mechanism, particularly in case of bacteria, an RNA hairpin followed by several uracil residue terminates transcription. As you can see, this is the growing RNA chain. So we have many residue nucleotide in this side, the 5' end. So in the 3' side, because of the specific sequence present at the 3' side of this RNA, so they make some intra-strand base pairing, as you can see here that GC, so they will make some hydrogen bonding. So this is intra-strand base pairing, and this is the hairpin structure or stem loop structure. This is the stem and this is the loop. We say this is stem loop structure or hairpin like structure. But this is happening because of a specific sequence present at the end of the gene. But this will destabilize the complex. Mechanically, it will inhibit the progression of RNA polymerase through the gene. If I try to show here something like this, so I would say the transcription is happening here. So if I say this is 5', 3', 3', 5' of DNA, and RNA is being synthesized. Now, if this stem loop structure, like here Watson-Crick base pairing, like intra-strand base pairing between GC and AT, not T actually, it should be A and U. So now, this structure actually destabilize this complex, and it will inhibit the progression of RNA polymerase is present here. This is our RNA polymerase. So it will inhibit the progression in this direction. So as a result of that, finally this RNA will be dissociated from this DNA, and we will get the RNA. So as a result of that, transcription will stop in this way. So now if you see carefully, following this stem loop structure, we have multiple U residues here in mRNA. Now if we have multiple U residues in mRNA, that means in the template we have multiple A residues present. So as a result of that stem loop structure followed by multiple U residue, that means that template and the nascent RNA, they have Watson-Crick base pairing. If you have now U on mRNA and A in template strand, that means you need less energy to dislodge this nascent mRNA. So as a result of that, I would say that stem loop structure followed by multiple U residues even help removing this nascent RNA from this transcription complex. But scientists observe that when they are trying to do transcription, they observe that in vitro transcription means we can carry out transcription inside tube also. So we just need some template DNA and some RNA polymerase and raw material ribonucleotides, and we can start transcription, right? RNA polymerase should be there. So now, while they were trying to synthesize RNA in vitro, they found that sometimes the product, the transcript particularly, is much longer than the gene itself. Then why it is happening? If all gene, they have some specific sequence at the 3' side which can make this RNA hairpin structure, then the length should be corresponding to the length of the gene. But they found some products, some transcripts are much longer, and they found that this is not only the mechanism. Another mechanism exists inside the cell, inside bacteria, that is called Rho-dependent transcription termination. Rho is a protein which requires some energy, it recognizes some sequence, and then this Rho protein, it's a complex protein, so it will migrate and finally it will dislodge this nascent RNA or the mRNA from the transcription complex, right? But during in vitro transcription, it is happening inside the tube, and we are not adding any other protein, no Rho protein there. That's why the length of those RNA are much bigger than the gene itself. So as a result of that, those are the two major things in case of prokaryotic transcription, where hairpin structure or stem loop structure as well as rho protein stop transcription. Now, in eukaryotes, the overall mechanism, like I always say, the fundamental mechanism is same, but little bit of variation. In eukaryotes, we have some more complexities, that's all. So, as you can see here, that eukaryotic transcripts need to be processed. Transcripts means the RNA, particularly here we are considering mRNA need to be processed. Like as you can see that the RNA, while it is being synthesized, the 5 prime end of this nascent RNA, nascent mRNA, messenger RNA, like is attached to some kind of modified nucleotide, like here modified like guanine residue, that is called 7-mg. Some methyl group is attached to this G residue, 7-mg cap, should be added at the 5 prime end. And then when the transcription is going on, some particular sequence, like for example here I mentioned here AAU, AAA, this sequence will recruit some enzyme called ribonuclease. From the name again you can understand that it will cleave RNA, like ribonuclease. So, it will cleave RNA. So, ribonuclease will cut this RNA here. So, as you can see here, and followed by some special things happen in case of eukaryotic mRNA, that one enzyme is called poly-A polymerase. From the name itself we can say what is the function of this enzyme, polymerase, but it is poly-A polymerase. So, as a result of that, it will add only A residue at the 3 prime end of this mRNA. So, that is why this is called poly-A polymerase, and this is template independent. So, that means in the template we do not have many corresponding residue, complementary residue present, not like that. This is template independent synthesis. So, it adds poly-A tail at the 3 prime. So, generally the length of this poly-A tail is 100 to 250 nucleotide long, poly-A tail. So, all these things like 5 prime cap as well as poly-A tail, it increase the stability of mRNA as well as it also because of this is more effective template for translation, which I will discuss during the translation. And now here if we see eukaryotic transcript also need to be processed. So, here when we are getting the mRNA from this gene, so this is DNA, this is the gene, beta globin gene for example here we are mentioning, and this is RNA. Here as you can see we have the 5 prime cap and 3 prime poly-A tail, and this is called primary transcript. It is not yet processed, means some portion of this gene which is called intron. So, this is intron, right, this is intron, it should be removed. So, finally we will get something like this. This is the mature RNA. So, this is the processed RNA where these introns are removed. This process is called splicing. Splicing is also required for eukaryotic transcript to make the eukaryotic mRNA, and that will be used for the translation in the next step. So, as a result of that, few things are different in prokaryote and eukaryote. Particularly if I say about the RNA polymerase, in case of eukaryote we have different types of RNA polymerase and they have different role, like RNA polymerase 1, 2, 3, but in case of bacteria or prokaryote we have same type of RNA polymerase and it transcribes all different types of RNA including mRNA, rRNA and tRNA. And also if we see here that the processing part is really different in between prokaryote and eukaryote. Like in case of prokaryote, no, those additional steps are not there, but in eukaryote we need to have this processing steps, the addition of 5' cap, 3' poly A tail, as well as removing the introns from the mature mRNA. And I should also mention the location of this transcription. So in case of prokaryote, as we know, we don't have the nucleus. So there we have just one compartment. So as a result of that, transcription is happening there, right? But in case of eukaryotes, in case of human cell, we have the nucleus, and all of us, we know that DNA is present inside the nucleus. So as a result of that, transcription as well as replication, these two steps are happening inside the nucleus, but translation is happening outside the nucleus. So whatever I told, this mature mRNA, after processing, it should come out from the nucleus into the cytoplasm in case of eukaryote, then it will be translated into protein, which I will be discussing in the next class. And that's all about this class, and you can refer these books for additional reading. That's all.